The second award is for basic research. So we have actually an introduction from Bob Langer through video.
It's a pleasure for me to be able to introduce Dr. Molly Stevens, who's won the Society of Biomaterials award. I know Molly very well. She was a postdoc in my laboratory and did an absolutely fantastic job. And she's also just a wonderful human being. She's been a great mentor, a great scientist and engineer in every way. Some of the scientific contributions Molly's made have been in all aspects of biomaterials. Some of them have involved new tissue engineering approaches, such as creating a bone bioreactor that you could actually use in vivo, creating also new kinds of new nanomaterial biosensors. So many companies have licensed her work, and she's been involved in getting companies off the ground, and has just done a terrific job. Molly's actually won all types of awards as well prior to winning the Society for Biomaterials Award today. Among those awards, she's actually, I think, the only person that's been elected to all 5 UK academies. She's the youngest person actually in history that's been elected to the Royal Academy of Engineering, and won numerous other awards for being one of the most outstanding scientists and engineers in the world under the age of 40. In summary, I think that she's just an amazing person who's just done great work in every way, and it's just an honor for me to have seen what she's done, and I know you're going to love hearing what she has to say to you today. It's a pleasure to introduce Dr. Molly Stevens. Thank you.
Molly, will you please come up?
Okay, perfect. So thank you so much for coming along, and thanks to Bob, obviously, for doing such a wonderful introduction. I absolutely loved being in his lab for my postdoc. So I'm going to tell you a little bit today about some of our work in the design of biomaterials for different kinds of applications. We work a lot in the field of designing materials for biosensing and also for regenerative medicine. As you'll have noticed, the award that I got is actually for basic research, and I think that comes from the fact that we do make these materials for applications, but actually we spend a lot of time on the fundamental studies as well. So really exploring that biomaterial interface, understanding it better, and thinking about how we can design it. So these are just some overview of some of the areas in our group. It probably looks quite different to you. It's quite a broad amount of research that we do within the group, but really it's all about understanding that biomaterial interface. And today I'd like to give just a few examples of some of the work we've been doing in that area with our wonderful team of researchers. So in terms of the regenerative medicine side of things, in this meeting that we're at today, Society for Biomaterials, you'll see there's a very wide range of scope of different types of material systems you can design, and some of them can be very simple and others can be more complex. And within our lab, we work on different gradients of complexity within materials. And it's worth pointing out that some of the more complex challenges obviously can be very exciting scientifically, but we also need to think very carefully about making sure we design materials that can get into patients. And sometimes even very simple material systems actually can have a great role in moving forward to clinical use. So I wanted to start just by giving 2 slides really on the work that first got me into this field of designing biomaterials for regenerative medicine, because my PhD was actually in single molecule biophysics until I went to MIT. And when I was in Bob's lab at MIT and together with Prasad Shastri as well, we designed this great new approach to regenerating bone where we actually used very, very simple biomaterial system, but we used it in a very nice way such that it could be injected under the periosteum as a liquid and then could cross-link in about 30 seconds into a rigid gel, and then could create an artificial cavity into which those stem cells from the periosteum could all proliferate and form lots of really nice new bone that you could use for transplantation. And this is just from that early work, from that postdoc time, where you can actually see, this is obviously an in vivo study, where you have the bone marrow in the middle of the leg here, the original bone, and then hopefully you can see that demarcation. This is all new bone that's formed to the left of the original bone within that artificial in vivo bioreactor. And by changing the biomaterial composition just slightly, so changing it so it didn't have calcium and also so that it wouldn't allow blood vessels to grow, we were also able to generate 100% cartilage instead, without having to add any growth factors or anything too sophisticated. The issue with this work, or one of the limitations of the work, is that you have to grow the bone in vivo and then transplant it. So you're going to have an incubation period of probably 6 weeks or so. So it's going to work for some applications, but not for others. So we've worked on many other material systems over the years, and you just heard a minute ago about the wonderful Larry Hench, and I wanted to just pay tribute to Larry a little bit because we worked together at Imperial College, and he was a wonderful friend and mentor, and actually he won this very same award, the Clemson Award for Basic Research, back in 1977, so it's a double honor for me in a way to be awarded this, knowing that Larry was so proud of having been awarded this many years ago. And Larry, as many of you will know, worked for a very long time on inventing and developing bioactive glasses. And some of the work that we took on in our group, together with other colleagues at Imperial College, was to look at some of those bioactive glasses and introduce different things into them. And one of the things we introduced that worked rather well was the introduction of strontium that then could have an effect on promoting osteoblasts to make new bone, but also on down-regulating osteoclasts that would be resorbing the bone. And this material worked very well in vivo, and we have a number of papers on that. But one of the things we got very interested in, and again comes back to this fact that we take the sort of fundamental side of the research very seriously, is that we didn't fully understand the role of the strontium. And so Hélène Hautefage, a postdoc in the group, did some beautiful studies much more recently. This was just published last year, trying to really understand all the different gene pathways that would be activated by the incorporation of strontium into the materials, and discovered some very, very interesting things to do with the fact that a lot of these sterile biosynthetic processing pathways are very upregulated in this material. So it's really about designing the material system, but also trying to understand it as much as we possibly can so we can design it even better. Now that takes us really to this more fundamental side of our work, which is all about understanding where biology and where cells can interface with materials. And of course, we're using that for our tissue engineering and also for any biomaterial implant that you want to put in the body. But it's also relevant anytime you want to understand about native tissue structure and also diseased tissue. And we work a lot in that area as well. So I just want to show you one more technology that we've been applying to cells and tissues. And this was some work that was done in collaboration with HMRI University. This is by Ciro Ciappini, who's a postdoc in the group. And we developed some nanoneedles that could interface directly with cells and with tissue. And these are very exciting because if you put cells onto them, the needles will spontaneously go into the cell. And similarly, they can spontaneously interface with tissues. And these are made of porous silicon, and you can just see here, you can make surfaces coated in many of these. You can change the spacing, you can change how pointy they are, you can change how quickly they degrade, all sorts of exciting things. And if you put cells on them, they start off rather rounded, you can see that here, but over time, the cells will spread out, and the needles will actually go here and interface very, very closely with the cell cytoplasm. And you can see that even nicer here in this cross-section, where the needle is shown in brown, and the cell cytoplasm in green, and the nucleus in blue, so they don't penetrate the nucleus. But as you can see, we have this very close interface now between this artificial needle and the cell cytoplasm, and actually you can hopefully see that porous structure. So that's extremely exciting, because what it means is we have a sponge-like structure that you can insert into the cell at hundreds of needles per cell. And that sponge-like structure that's interfacing with the cell means you can do two very, very cool things. You can monitor things going on within the cell, but you can also deliver to the cell. And so, for example, you can put cells on these. These are some cancerous cells from the esophagus, and you can monitor pH all over the cells, but you can also monitor enzyme activities, and we've published in those different kinds of areas, and also applied these to human tumor tissue to be able to map the interface between the cancerous and the tumor tissue areas. In terms of delivering in vivo, as would be most relevant for the tissue engineering, we did some in vivo models at HMRI. Well, the team at HMRI did the in vivo models. And what we did was, first of all, deliver to both skin and to muscle using the nanoneedles. This is actually just a picture of a fluorescent dye here. And then moved on to thinking about delivering things that would be very, very interesting biologically. So we all know, for example, that often when you implant a scaffold that you've engineered in vitro, in vivo, it might die because you can't get blood vessels to grow in fast enough. So we posed the challenge of, well, can we pre-vascularize a site using this nanoneedle technology? And in fact, we can do that very nicely by delivering to muscle a plasmid DNA that encodes for vascular endothelial growth factor. And this is just the control measurement here. After 2 weeks, the muscle looks like this. But if you deliver just really very quickly, so in less than a minute, the VEGF plasmid DNA with your needles, you can see all these new blood vessels will start to form. This is after a week, and then after 2 weeks, they start to mature, and you can see that blood vessel maturity over time. And this is really just an example of a platform technology that you could use to deliver all sorts of other plasmids for other growth factors and other kinds of molecules. So that's all I wanted to say really on the tissue engineering side. I want to just tell you a couple of things about what we've been doing in terms of thinking of materials for biosensing. I'd say probably half our group is working in this area. And we tend to think of, particularly actually tend to work on nanoparticles for different biosensing applications. And the way we think about engineering these is to consider, obviously, the core of the material that's going to be very important for material properties, but also how we functionalize the surface of that. So this is really key to all sorts of biomaterials design, and how we then make that surface very active so it can be used for different biosensing applications. And we look at these for applications in cancer and also in infectious disease and in global health. So if you think about functionalizing the surface of these nanoparticles, you can actually do all sorts of really amazing things now with the different chemistries that we have available. And we're particularly interested in how we can use specific biomolecular interactions to change the readout of our nanoparticle systems so you can do some really sensitive biosensing. And I'm just going to show you just a couple of examples of the work we've been doing in that area. So firstly, we got very excited actually about how we might be able to use enzymes to control the way that nanoparticles can grow and how we could couple that to biosensing. And so Roberto de la Rica, who's now a professor in Scotland, used enzymes coupled to a biosensing mechanism to control whether nanoparticles would grow as dispersed, non-aggregated gold nanoparticles, or whether they'd grow aggregated and a much more ill-defined type of architecture. And if you do that change to those nanoparticles right down at the nanoscale, you actually end up with a readout you can detect with a native eye. So those dispersed particles will look red in solution to our naked eye, and the aggregated ones will look blue. And so your human eye can actually detect this change going on, and it correlates to a biosensing that is many, many orders of magnitude more sensitive than what you would get with a traditional ELISA-based protein sensing. Similarly, we've used these kind of nanoparticles for many other applications. I'm just going to show one, well, maybe two last applications here. We've been very interested to see how we can use them to detect important enzymes called phospholipases. These are important in all sorts of diseases. I've just listed some of those there, but also cancer and other diseases. And they're enzymes that will cleave through phospholipids, and they're very, very sensitive to how the phospholipids are presented. So in the top right, you can see an image from Herman Gaub's group where you have a phospholipid bilayer on a surface, and then hopefully you can see these little tracks. These are little tracks where individual enzymes have been cleaving through the phospholipids. But they're normally detected with strategies that involve chemical modification to the lipid substrate, and so you're not actually getting a readout of real enzyme activity. So what Daniel Ali did in our group, he's now professor in Linköping, was to develop an assay where we could use a native lipid substrate, so we could get a real readout of enzyme activity. And the assay looks like this. We actually start off with some gold nanoparticles here in solution, and then we have a cross-linker peptide within a liposome structure. And if the enzyme phospholipase A2 is present, and you can see it would cleave the phospholipids here, this liposome structure will disrupt, these guys will leak out, and they will form these 2,4 helix bundles, cross-linking structures that bring the particles together. And so now your solution is going to change from red to blue yet again. And you can get out dose response curves for that, and it's a really nice piece of work from Daniel and other members of the team. Now, to get this into a more user-friendly format, Rob Chapman, who's now a career development fellow in Sydney, worked on this very hard to get this working in a point-of-care format, so within a lateral flow device. And to do that, we had to work on the system quite a bit, really think about the valency of the cross-linkers that we want to use, the different charges, the amount of gold, and many other parameters that were optimized over time and published. And the end result was that actually you can end up with an assay that's extremely sensitive for acute pancreatitis, but also for rheumatoid arthritis. And this has now been tested in many, many hundreds of patient samples with our clinical collaborators in London. Now, the very last thing that I want to show you is some recent unpublished work. Just one last slide on that, which is some other research that we've taken on our gold nanoparticle-based biosensing and lateral flow format that we've now taken out to Africa. So this was tested in 120 or so Ebola survivors and control patients in Africa to map the serological surveillance of patients that had had Ebola. And we've coupled this to an app that we've developed with a smartphone in Uganda. And we were able to determine with great sensitivity and specificity what strain of the Ebola outbreak these patients had. And I think this is extremely interesting as a technology because you normally are only able to do these kind of measurements in a specialized lab. So this is the first point-of-care device that's been used in the field for these kind of measurements and really just takes not very long at all to do. So this is unpublished, and hopefully we'll be submitting this very soon for publication. And so it just remains for me to really thank my wonderful research group, who are just terrific in every single way, and all the mentors that have been so inspiring and helpful to me over the years. There are very many, my PhD supervisors, of course, postdoctoral advisors, and many of the other less formal mentors, but that have also played an extremely important part in my life. And I would just like to also acknowledge our funding sources and the collaborators for the data that I've shown here. I've had people's names on every slide for the data I've shown today, and of course there are many, many, many other fantastic projects and fantastic people that have helped us that unfortunately I'm not able to do justice to here, and many of our group, of course, attending the conference as well, and I've just highlighted them there. And thank you so much. This is a massive honor for me. I'm really tremendously honored to get this award and appreciate it very much. Thank you.